photosynthesis, Purpurbakterien, Van Niels' isolation technique, anaerobic pure culture. From a raw culture of various Purpurbakteria, Van Niel was the first to isolate an individual strain. One of his pupils, Professor Norbert Pfennig, demonstrates the pure culture technique developed for that purpose. The principle is the serial dilution of a drop of the mixed culture. In this way, the concentration of bacteria in suspension is progressively decreased. With each stage of dilution, the distance between individual cells becomes greater, until finally, only a few bacteria are distributed throughout the glass tube. The liquefied agar used as a medium requires rapid manipulation. When too hot, it damages the bacteria. When below 40 degrees, it hardens. After it sets, it's coated with paraffin as a protection against oxygen from the air. We are dealing with oxygen-sensitive organisms. The bacteria within the hardened agar are now in an anaerobic milieu. For Purpurbakteria, an illuminated incubator, Van Niel called it the light cabinet, has to be used. Within a week, the cells from the serial dilution grow to visible colonies. Originally, the glass tube was broken to obtain an individual colony. It is more elegant to carefully remove the paraffin and, with a capillary, inject sterile pressurized air between the agar and glass wall, thus pushing the plugs out of the tubes. The desired colony can then be sucked up into a sterile capillary. These cells are suspended in a droplet, which is then subjected to a second serial dilution. Only after a second isolation procedure can one be sure of obtaining a genuine pure culture with sufficient certainty. Promoted by sulfide and bicarbonate in the agar, the Purpurbakteria grow into homogeneous individual colonies which are inversely proportional in size to their total number present in the tube. The hardened agar only serves to isolate the bacteria. After the pure strains have been obtained, liquid culture in airtight, closed flasks is suitable. Since 1930, Van Niel and his school were thus able to isolate numerous strains of Purpurbakteria and to breed them in large quantities.
Plasmodium falciparum, invasion and multiplication in erythrocytes. The malaria parasite Plasmodium is transmitted to humans by the female Anopheles mosquito. In penetrating the skin, the organisms are inoculated into the bloodstream with the insect's saliva. There they multiply at first in liver parenchyma cells and later in red blood cells. The invasion of these host cells by Plasmodium is a highly complex process which can best be investigated on the parasitic development stages in the blood itself. The following description is solely concerned with these stages. The infective form specific to the red blood cell is the merozoite, a 1 to 2 micrometer long ellipsoidal cell with a conical tip. It has a basal nucleus. In the apical region, it has club-shaped secretory organelles, the rhoptries. Their common secretory duct emerges at the apical pole of the merozoite. Further organs of the parasite are the filamentous micronemes and the dense granules. Together with the rhoptries, they act during the penetration process in the invasion of the host cell. The surface of the merozoite is covered with 10 nanometer long filaments, the so-called brushy coat. Its host cell, the erythrocyte, is contacted by chance. This contact causes a slight deformation of the host cell's membrane. Invasion is only successful when the merozoite has attached itself to the erythrocyte membrane by its apical end. Apical contact triggers the secretion of lytic substances from the rhoptries. Secretion continues throughout the entire invasion process and causes invagination of the erythrocyte membrane. The enzymes released by the micronemes contribute to this invagination. During penetration, the ring interface zone between the host cell and the parasite is continually moving to the posterior end. The surface coat of the parasite is shaved off level with the junction and serves as an additional seal for the invasion chamber. After the completed entry, the merozoite is now fully enclosed in the invaginated erythrocyte membrane. This enclosure is termed the parasitophorous vacuole membrane. Shortly after internalization, there follows the exocytosis of the dense granules. Their proteins are transported into the cell interior and to the membrane enclosing the parasite vacuole. Once the parasite has been internalized in the host cell, the invasion process is completed. After flattening and vacuolization, the merozoite attains the typical developmental stage known as the ring form. The interaction between Plasmodium falciparum merozoites and human erythrocytes can be studied and recorded using in vitro cultures. Chance contact gives rise to a slight deformation of the erythrocyte surface, which may last for 9 to 15 seconds. This may be initiated by the difference in the surface charge of the two cells, the negatively charged erythrocyte and the less negative merozoite membrane. Once attachment has been established between the apical complex and the host cell membrane, the actual invasion process is initiated. In vitro studies show that it lasts up to 15 seconds. The parasite is now hardly distinguishable inside the red blood cell. Shortly after invasion by a merozoite, the host cell crenates, caused by hygroscopic interactions inside the cell. This process is reversible. Only a few minutes later, the erythrocyte has resumed its original biconcave shape. Recognition receptors on the host cell and the parasite play an important role in the invasion process. the Merozoite surface antigen becomes effective immediately after first contact with the host cell, while at a later stage, the erythrocyte binding antigen at the apical complex of the Merozoite is responsible for conditioning the erythrocyte to invasion. Important receptors for the Merozoite proteins include the glycophorins A, B, and C on the surface of the host cell. Using the electron microscope, it is evident that fine fibrils extend between the apical Merozoite pole and the surface of the erythrocyte, inducing the invasion process. Also, in later stages of invagination, the Merozoite remains in contact with the vacuole membrane by a narrow band which is continuous with the common duct of the rock trees. The circumferential attachment zone between the parasite and host cell is marked by membrane thickenings, which are particularly well recognizable in the magnified insert at bottom right. The junction zone is continually displaced to the rear until the invasion orifice is finally closed by fusion at the posterior end of the Merozoite. Here, invasion is practically concluded. Inside the erythrocyte, multiplication of the parasite by asexual fission or schizogony is now initiated. Soon the young Merozoite is transformed into the signet ring stage. The characteristics of this development stage are a loop of thin cytoplasm, a vacuole, and a marginal nucleus. In the case of multiple infection, the ring shape is not recognizable with any certainty. The trophozoites, here a double infection, are amoeboid forms containing diffusely distributed grains of brown pigment. Further division of the trophozoite leads to a schizont. Mature schizonts occupy most of the red cell and have a mulberry-like surface containing between 16 and 32 merozoites. In vitro, the merozoites are released 40 to 48 hours following invasion by rupture of the host cell. Together with the merozoites, the metabolized hemoglobin, the hemozoin or malaria pigment, is set free into the blood plasma. In contrast to in vitro cultivation conditions, the erythrocytic cycles in the human organism run in synchrony. The simultaneous release of all merozoites and pigment causes the typical attacks of malaria fever. The invaded erythrocyte has lost its discoid shape. Shortly before burst, considerable changes in the cytoskeleton have occurred, depriving the cell of its capability for self-deformation. The cell membrane becomes unstable and easily ruptures. The released merozoites can immediately initiate a new erythrocytic cycle. The invasion of erythrocytes by Plasmodium merozoites is a complex cellular, biochemical and molecular process in a highly specific host-parasite relationship. The molecular elucidation of this mechanism is a vital key to the development of a vaccine as well as to the recognition of new therapeutical strategies.